Are we good to go? Can everyone see? Okay. Let's go. So, 6.5 Neurons and Synapses. Oh my god. Hi, Chase. Okay. 6.5 Neurons and Synapses. So, first of all, we need to know what neurons are. The nervous system is composed of cells called neurons. So neurons are the basic cell part of our nervous system. These cells carry messages at high speeds in the form of electrical impulses. So the way your entire nervous system works is through electricity and electrical impulses. Many neurons are very elongated and they carry impulses for long distances. Carry impulses across long distances. I might as well fix this now. Across long distances in a very short time. So that basically means that there's only so many neurons that are involved in our nervous system, but because they're so elongated, they can carry impulses very quickly. Myelinated nerve fibers that have a myelin sheath around them with small gaps called nodes of Ranvier allow the nerve impulses to jump from node to node. So in the structure of a neuron, which by the way you do need to know, there's a nucleus. I'm going to change the way I'm streaming so that I can point, because I can't do that if I'm not showing my entire screen. I'm going to do that again. Okay. So there is a nucleus. This entire thing is the cell body, which has the cytoplasm. And then the cell body branches into dendrites, which are what make connections to the axon terminals of other neurons. So that's the main part of a neuron. And then the neuron then is very elongated, and this nerve fiber is called the axon. The axon is covered by the myelin sheath, which is basically just cells that cover the sheath for ease of electrical conductivity, because the entire neural network, or the axon or the nerve fiber, has charges, but with the myelin sheath it basically makes it easy for the impulses to jump from one place to another instead of having to travel through the whole thing. And that happens because of the nodes between each cell or each section of the sheath, and those spaces where the sheath does not cover the fiber are the nodes of Ranvier. And then those nodes are where the impulses jump from, for example, from this node to this node, and then to the next, and then to the next. So it doesn't have to travel the entire length of the neuron. It just travels from node to node. The entire nerve fiber is called the axon, and then it ends in axon terminals, which then either attach to another the dendrites of another neuron, or they'll attach to skeletal muscle fibers, or they'll attach to any muscle, any other neural network. So saltatory conduction is what speeds up transmission. Saltatory conduction is when the nerve impulses jump from node to node between the nodes of Ranvier and between the myelin sheets. Does that make sense? Did everyone get that part? Yeah, no worries, Storm. This is being recorded, so if you ever want to look at it again, you can. Feel free to later on. Okay. The next is synapse. I'm Batman now. Okay. The next is synapses. So a synapse is the junction between either two neurons or between a neuron and a receptor or effector cell. So if you look back here, basically where the according to this diagram, where the motor end plates or the axon terminals are, between that and either another neuron or that and the actual muscle fibers, that's where synapses are. So they don't actually physically connect. There's a gap. That's where the junction exists. So plasma membranes of neurons are separated by a narrow fluid-filled gap called the synaptic cleft. The synaptic cleft is the space between these two neurons. Hold on, I'm going to change this again just because I feel like it's easier when I can point at stuff. Okay, so basically this is one end of, this is probably, yeah, because this is the presynaptic neuron. So this is the neuron this is the axon terminal of a neuron, or the motor end plate of a neuron, and this is probably one of the dendrites of the other neuron. So between these two, this is obviously, there's a plasma membrane here, but between the synaptic knobs, which is labeled here, between these two there's a space, and that's called the synaptic cleft. This entire thing is the synapse, but this space specifically that's really narrow and has fluid in it, that's called the synaptic cleft. So because of synapses existing, messages can be passed from one neuron to another, or from one neuron to the receptor or effector cell. So there's always neurotransmitters that messages that are passed across synapses are in the form of chemicals, which are the neurotransmitters, and the neurotransmitters will always pass from the presynaptic neuron to the postsynaptic neuron. So in this case, this synaptic knob is part of the presynaptic neuron, since this shows an arrow going downwards. That's where the nerve impulse is traveling, and this bottom one is called the postsynaptic neuron. So the way messages travel through synapses. The first one is that the nerve impulse will reach the end of the presynaptic neuron. So if we go back here, basically it'll travel through the entire, it'll do the saltatory conduction and reach the end of the axon terminals, or the motor end plates. And then once it reaches here, that's step one. After it reaches there, the neurotransmitters will basically be contained in vesicles, are contained in vesicles for transfer, and then they will attach to the membrane of the presynaptic neuron and open up and release their contents, the neurotransmitter, into the synaptic cleft. So, and then the third step is that the neurotransmitter, of course, will diffuse through, because it's put into the entire synaptic, all the neurotransmitters are diffused are left open into the synaptic cleft. Then they will diffuse across the synaptic cleft and bind to the receptors on the postsynaptic neuron. So after they bind to the receptors will receive the message or the information that they're trying to transmit, and then the nerve impulse will set off along the postsynaptic neuron, and then basically this would be a dendrite, so it will go through the entire process and through the saltatory conduction again, but it happens after the neurotransmitter has been recepted by the receptors over here. There's also depolarization. I missed that step in the middle of this. So sodium ions will from an increase in the concentration between the synaptic cleft, sodium ions will enter the postsynaptic neuron, cause depolarization, and therefore will result in the nerve impulse being passed through the entire neuron, which we will discuss later. Depolarization and repolarization is something we will discuss later on. Yeah, so then the nerve impulse will be set off because of depolarization. And then the last one is the neurotransmitter will be broken down within the cleft, and then will get reabsorbed into vesicles. Do we understand synapses? Are there any questions regarding synapses? Really? Okay, I will try that. I will try that after Oh, it does. Can you see it though? Okay, that's lovely. Okay, I'll do that. I'll do that after that. -50, -30, -70. Yes, we will talk about that. By the way, Lipid, I love you too. Love, love. I will change my name for you. After. Anyway. I will. Shh. Anyway, before we talk about resting potential and action potential and all of that fun stuff, firstly we need to know about cholinergic synapses. So not all synapses will use the same neurotransmitter, but many of them use acetylcholine or acetylcholine, choline. Now I don't even think it's a word anymore. But the synapses that use acetylcholine or choline are known as cholinergic. I can't pronounce this anymore, guys. I literally can't pronounce this anymore. Cholinergic. Whatever. You know what I'm talking about. Those synapses are the ones that use acetylcholine. So the presynaptic neuron secretes acetylcholine into the synaptic cleft, which diffuses across the synapse and then binds to the receptors in the postsynaptic membrane. So the same process of synapses occurring will happen, but in this case, it just happens with just acetylcholine. The acetylcholine is then broken down in the synaptic cleft by the enzyme cholinesterase. I can pronounce this, I can't pronounce that. That's funny. Cholinesterase, which produces acetyl groups and choline, which the choline will then be reabsorbed by the presynaptic neuron to make acetylcholine again. The next is neonicotinoid pesticides. So this is an example that you need to know, and this was, I think, a question that I tried to answer the other day because we were talking about neonicotinoid effects and things like that. So neonicotinoid pesticides are what bind to acetylcholine receptors in the postsynaptic membranes of cholinergic synapses in insects. So what happens is, if you look at the receptors over here, these are the receptors that accept the messages from the neurotransmitters here. But then these receptors are bind with neonicotinoid pesticides and prevent the binding of the neurotransmitters. So what happens is, obviously, since they bind to the receptors, they can't then be broken down because cholinesterase is not eligible to break down neonicotinoid. And then since it doesn't get broken down, they remain bound basically forever. So then there's no acetylcholine binding, and then that basically stops the neural network, or like the flow of the impulse. And then they just block all synaptic transmission, which ultimately will kill the insect. So that's why it's such an effective pesticide, because it basically just attacks the brain of the insect and then therefore stops it from functioning altogether. But the unfortunate circumstance of this is that other organisms are also affected, not humans, but other insects or other organisms are also affected because of their action on acetylcholine receptors. So unfortunately, honeybees, which are a really good pollinator, are also killed along, and they're not the intended target. So that's the negative of neonicotinoid pesticides. I'm really enjoying this pointer, if you can't tell. Okay, now we go into the entire process of basically this the entire, what's it called? Nerve impulse. Yes. So, first we need to know what a resting potential is and then what an action potential is. So, a resting potential is the voltage across the plasma membrane of a neuron when it's not conducting a nerve impulse. So, that's the voltage that a neuron will maintain when it's not conducting an impulse. So within the plasma membranes of axons, there are sodium-potassium pumps. So they will pump sodium out and potassium in by active transport. And concentration gradients of both sodium and potassium are established across the membrane, which allow the active transport to occur. That occur due to active transport, and then they will cause the voltage to exist because a concentration gradient exists. And sodium and potassium ions are used within the pumps, and therefore they form charges, relative charges between the inside of the neuron and the outside of the neuron. So at resting potential, the inside of the neuron has a net negative charge compared with the outside because it has chloride and other negatively charged ions. This has nothing to do with the sodium and potassium yet because they're at equilibrium at this point, but the net negative is established by chlorides from chlorides and other negatively charged ions. So there's obviously, because there's a net negative charge within the neuron, there is a voltage across the membrane because there's a potential difference across the membrane. So the potential difference at the beginning is negative 70 MV, which is, I believe, microvolts. Correct me if I'm wrong. But yeah, so it's at negative 70, which is something you just need to know. That's what a typical resting point for most humans would be. Action potential. Now, action potential the action potential is a whole process, which is the depolarization and repolarization of a neuron due to facilitated diffusion of ions, in this case sodium and potassium, across the membrane through voltage-gated ion channels. This is a mouthful, but you need to know what an action potential is. So basically, since the resting potential is at 70, if the potential across the membrane will rise from negative 70 to negative 50, so it'll go up by a little bit, then the voltage-gated sodium channels will open, and sodium ions will diffuse into the nerve fiber. When that happens, since sodium ions are positively charged, Na+, they create a net positive charge within the neuron as compared to the outside. So since there's more sodium ions within the inside of the neuron, there will be a net positive charge, and there will be a potential difference between the membrane, which is reversed as compared to the resting potential. Because in the resting potential, the inside has a net negative. At action potential, at depolarization, at the stage of depolarization, the inside will have a net positive, while the outside will, as compared to the outside. But then, at the same time, there's a reversal of the membrane polarity, the reversal of the membrane polarity since depolarization occurs. This reversal will cause the potassium channels to then open, allowing potassium ions from within the cell to diffuse out, which are also positive, to diffuse out and go down the concentration gradient. And then that will cause the inside of the neuron to develop a net negative charge, again, as compared to the outside. The potential across the membrane will be restored to the resting potential, and this is when repolarization happens. So if you look at the entire graph of the process, this is at resting potential, it's at negative 70. this is when it reaches the threshold potential. This rise is when this rise happens, this is when the sodium channels will open. Because the sodium channels open at this point, more sodium ions come into the neuron, which means that the potential energy, the inside of the neuron will turn into a net positive charge, and then that will cause it to go up. Obviously, here it's at positive 30, it'll go up really high, and then so this entire process is called depolarization. And then once it reaches a certain threshold, once it's up way above, and this reversal will cause the potassium channels to then open and the sodium channels to close, which means that the potassium ions will see a sudden fall from within the neuron, and they will move out, and then that will cause repolarization, which makes the net charge of the across the membrane go further down than the resting potential, but then it stabilizes itself when it comes back to the resting potential because of the closing of both the channels. Could they ask us to draw the graph? It could. They could. I think they could. I don't know, but I would guess they probably could. Okay, it's millivolts then. Sorry, my bad. 2 was depolarization. Yes, 2 is depolarization. So 1 is resting potential, and from resting potential to a slight increase in the potential. 2 is depolarization. 3 is repolarization. And then 4 is when it returns back to the resting potential due to the pumping of potassium ions out and sodium ions out and potassium ions back in. So because it drops so low, then it just has enough energy to activate the pumps, and then it'll basically pump. It'll return back to the original configuration of having potassium within the neuron and sodium out of the neuron. 3 is repolarization. Yes. Yeah, that's basically it. Well, is there anything else that I need to go through? Yes, there was this one diagram which I think I forgot to insert into the thing, so I'll insert it right now. But so there's basically a whole process of this is a copy of the same thing. There's a whole process of propagation that occurs basically where Neurons and Synapses is actually very short. Okay, so basically a nerve impulse obviously is an action potential that travels along the axon of the neuron from one end to another. So we know a nerve impulse will basically start somewhere here and then work its way through the axon until its motor end plates, and then either transfer it back to another neuron or transfer it to the muscle fibers. In which case, obviously, it'll just do the action that it's supposed to do. But let's go back. So a nerve impulse is the series of action potentials that gets traveled. There's an action potential whenever a part of the axon reaches the threshold potential. So whenever any part of the axon reaches this threshold level of -50, then it'll trigger the process of the action potential. The action potential only starts after the threshold level has been reached. So an action potential in one part of the axon will trigger an action potential in the next part. So this is where the propagation occurs. So this is at resting potential. This is at action potential. So when for example, it goes from this, where in sodium is on the outside, potassium is on the inside. This is what usually happens. When you go into action potential, the sodium gets pumped in and the potassium gets pumped out. That's how the entire process of the action potential happens. And while some of them will be restored back to this conformation, some of the sodium ions will be moving towards the next region. So obviously we're looking at a neuron that's functioning this way. This is its cyton. This is its cell body, and this is the axon terminals. And if it moves in this general direction, if this is an action potential, this region past these two pumps, for example, this will return back to resting potential, but some of the sodium ions will move their way to the next region. So by next region, I mean the next node. And when it moves to the next node, that's what triggers it to go to the threshold level. The only reason it goes from a resting potential to the threshold level is when from the previous action potential, some of the sodium ions make their way to the next region, and then therefore cause the threshold potential to be reached, and then trigger the rest of the action potential. So that's what happened. That's what the propagating is for. Yeah, that's it, I think. That's it for neurons and synapses. So if there's any questions, go away and take it away. And there's gonna be, yes. So sorry, I didn't talk about it. The oscilloscope trace is this graph. This graph is an oscilloscope trace. So the changes in our membrane potential can be measured using electrodes, and then the oscilloscope, which is this, is what shows where the where the changes occur. That's the trace. But yeah, so again, I'm gonna get more water because I've drank all of my water. But yeah, if there's any questions, I'm gonna drink more water and take a break and breathe. But if there's any questions, I'm willing to answer them. It is in the guide, but I think it's just a name thing. You just need to know that the trace exists. Like, it's called an oscilloscope trace. Do people want me to do 6.6 today? If not, then I don't have to do it today. I can do it tomorrow because I feel like a lot of people are leaving. So I can continue it tomorrow. I have no issues with that. Because I realize for a lot of people this is late. Yeah, all the guide says is that an oscilloscope trace exists and it can be measured using electrodes, and that's what the trace basically tells you the entire process of resting potential and action potential. So I don't think you have to stress about knowing it really all that much. But yeah, how are we feeling, guys? If this is too much information all for one day, I can continue it tomorrow, no issues at all, because it is late for me too, so I understand. It is a little after midnight for me too, so I presume we're in the same boat. Yeah, no, I will keep going if people are still engaged, but yeah. No, it's okay. I will sleep after. That's why I'm only doing 6.6, and then everything else I'm gonna do tomorrow. which is why I was asking. 6.6 is homeostasis, hormones, and reproduction. I believe the exact title is hormones, homeostasis, and reproduction. Like FSH.